There is an experiment that ecologists have thought about but never run, because the consequences would be catastrophic and irreversible. Remove all the flies from the earth, every species, every larvae, every adult, every egg, everything belonging to the insect order Diptera, 160,000 documented species, distributed across every ecosystem on the planet from tropical rainforest to Arctic tundra to the soil beneath your own lawn. Gone. The thought experiment does not end well. Within days, carcasses that would have been processed by blowfly larvae begin accumulating across every habitat on Earth. Decomposition slows. Nitrogen and phosphorus locked inside dead organic matter stop cycling back into the soil. Certain plant species, including some crops humans depend on for food, begin to fail for lack of pollination. The fish populations that depend on aquatic fly larvae as their primary food source begin to decline. Then the birds that eat those fish. Then the predators that eat those birds. The cascade continues for as long as you're willing to follow it. And all of it traces back to the removal of an insect that most people, in most moments of their lives, would pay someone to eliminate. This is the truth about flies, not the truth that makes them less disgusting. The truth that makes the disgust, by the end of this script, feel like a significant misallocation of your concern. Start with decomposition, because it is the role most people half know and almost nobody fully understands. When an animal dies, any animal, anywhere on Earth, the first organisms to arrive, in most environments, within minutes to hours, are flies. Specifically blowflies and flesh flies, whose sensory systems can detect the chemical signatures of death from distances of several kilometers. They arrive before almost everything else. They lay their eggs, and within hours, the larvae that hatch begin the process of breaking down biological material with an efficiency that nothing else in the ecosystem matches. A maggot mass working on a carcass is not simply eating. It is chemically accelerating decomposition in ways that took scientists decades to fully characterize. Maggot masses generate internal temperatures that can exceed the ambient air temperature by 10 to 20 degrees Celsius. A biological heat produced by the metabolic activity of thousands of larvae feeding simultaneously, which in turn accelerates the enzymatic breakdown of tissue around them. They produce digestive enzymes that pre-liquefy their food before consuming it, effectively extending their reach beyond what they physically contact. They fragment organic material into smaller and smaller pieces, dramatically increasing the surface area available for microbial action, which further accelerates the chemical decomposition the microbes are driving. The result is that a carcass processed by fly larvae returns its nitrogen, phosphorus, and carbon to the soil in days or weeks. A carcass in an environment where flies are absent may take months or years to reach the same point. Every nutrient cycle in every ecosystem on Earth, the cycles that feed the plants, that feed the animals, that eventually feed us, runs faster because flies are doing this work continuously, invisibly, in every corner of every habitat where organic matter accumulates. This is not a minor contribution to the planet's chemistry. It is load-bearing. Now consider pollination, because this is the role most people have never been told flies play. Bees receive the credit. Bees deserve significant credit. They are extraordinary pollinators, and their decline represents a genuine, well-documented crisis in agricultural ecology. But the narrative that centers bees as the primary pollinators on Earth is incomplete in ways that matter. Hoverflies alone, a single family within the broader fly order, are estimated to be the second most important pollinator group in the world, after bees. In multiple agricultural systems across Europe and North America, hoverflies have been found visiting more flower species than bees in the same habitat, and in some studies visiting flowers with higher frequency. They pollinate crops including onions, carrots, apples, mangoes, and stone fruits. They are the primary pollinators of some wild plant species. Not a backup option, but the only reliable option the plant has. And there is a specific ecological situation in which the fly's role in pollination becomes not secondary to the bees, but irreplaceable: cold. Above the tree line, in high mountain terrain, in Arctic and subarctic environments where temperatures are too low for most bee species to operate reliably, flies continue to fly. They continue to visit flowers. They continue to transfer pollen between plants that would otherwise have no pollinator present at all. The entire pollination ecology of certain mountain and Arctic plant communities is structured around fly activity, specifically because the cold that limits bee activity is not a limiting factor for flies in the same way. There are plant communities on Earth, specific documented ecosystems, where removing flies from the pollinator guild would not simply reduce the pollination rate, it would end it. There is a more specific and more surprising contribution that flies make to human food that almost no one knows about. Chocolate. The cacao tree, the tree whose seeds are processed into chocolate, produces flowers that are small, complex, and located close to the trunk in configurations that exclude most large pollinators by simple physical geometry. Cacao flowers require a very small pollinator that can navigate their structure. In the wild, and on cacao plantations worldwide, that pollinator is a midge, a tiny biting fly so small it is almost invisible to the naked eye. That is the primary natural pollinator of cacao. Without midges, cacao production drops dramatically. Plantations in regions where midge populations are healthy report better yields than those where midge habitat has been destroyed or reduced. The global chocolate industry, worth an estimated $130 billion annually, is dependent on the pollination services of an insect that most people, if they noticed it at all, would attempt to swat. Vanilla presents a similar case. The vanilla orchid's pollination in its native range in Mexico was primarily carried out by specific local bee species, but vanilla cultivation outside that native range has consistently struggled with pollination, requiring manual hand pollination at significant labor cost, specifically because the specific fly and bee species that pollinate it naturally are absent. The difference between vanilla that costs $3 for an extract bottle and vanilla that costs $30 is, in significant part, a story about which pollinators are present and which are not. Then, there is what flies do for medicine. Because this is the dimension of fly biology that requires the most dramatic revision of the popular image of what a maggot is. Maggot debridement therapy is a medical procedure approved by the United States Food and Drug Administration and practiced in hospitals and wound care centers in multiple countries. In the procedure, sterile blowfly larvae are applied to chronic, non-healing wounds, diabetic ulcers, pressure sores, infected surgical wounds, where they perform a specific function that no pharmacological treatment has ever matched in certain patient populations. They clean the wound. Maggots used in wound therapy produce digestive enzymes that break down only dead, necrotic tissue while leaving living tissue intact. A selectivity that surgical debridement cannot always replicate with comparable precision. They consume the dead tissue and the bacteria colonizing it, including antibiotic-resistant bacteria like MRSA that standard antibiotic regimens cannot eliminate. They produce ammonia that increases the pH of the wound environment in a way that further inhibits bacterial growth. And they stimulate the production of growth factors in surrounding healthy tissue that accelerate wound healing beyond what would occur without their presence. Maggot debridement therapy has shown documented success in wound cases that were progressing toward amputation, in patients who had exhausted other treatment options, and in infections where the specific bacteria involved showed resistance to every available antibiotic. The treatment was used in field medicine before antibiotics existed, fell out of use when antibiotics made it seem unnecessary, and has returned to clinical practice precisely because certain infections have become antibiotic resistant enough that the maggot is, again, the most effective option available. The animal that makes this possible is a blowfly larva, the same organism that processes roadkill, the same organism that appears on a carcass within hours of death, the same organism that makes most people feel sick to look at. Flies are also doing something in the criminal justice system that almost no other organism can do. They are solving murders. Forensic entomology, the use of insect evidence in legal proceedings, depends almost entirely on flies. The principle is straightforward. Blowflies arrive at a human body within hours of death in predictable conditions, lay eggs on a predictable schedule, and develop through larval stages on a temperature-dependent timeline that scientists can calculate backward from the developmental stage of the insects found on the body. The minimum time since death is established by determining the age of the oldest insects present. This calculation, the postmortem interval estimate derived from fly evidence, has been accepted in courts of law across multiple countries. It has been used to establish cause of death, to challenge alibis, to corroborate or contradict other forensic evidence, and to place specific individuals at specific scenes within specific time windows. It has helped convict killers and, in documented cases, helped exonerate the wrongly accused. None of this is possible without the fly's predictable, inexorable, rapid response to death. Its behavior, arriving within hours, developing on a known schedule, responding to temperature in a mathematically characterizable way, is what makes it useful. And what makes it useful is precisely the biology that makes it, in most human contexts, repellent. Here is the truth about flies, held clearly rather than softened. They are not pleasant. The housefly lands on food and on feces and carries bacterial contamination between them. Certain fly species transmit diseases that kill hundreds of thousands of people annually. The buzzing of a fly near food at a summer picnic is an accurate signal of a sanitation problem that should be addressed. None of that is untrue. But it is also true that every nutrient cycle in every ecosystem on Earth runs through fly larvae. That the second most important pollinator group on the planet is a fly. That a fly larva is an FDA-approved medical device for treatment-resistant wounds. That the most reliable method for establishing time of death in a murder investigation is built entirely on fly behavior. That without the midge, chocolate would be an agricultural footnote. The fly did not evolve to be disgusting. It evolved to do work, essential, irreplaceable, load-bearing ecological work, that happens to look, from the outside, like the most repellent thing in nature. We are alive, and fed, and cured, and sometimes vindicated in court, in part because of what the fly is doing while we are trying to wave it away. That is the truth you never knew. And it was always sitting right there, buzzing in your face, waiting for you to understand it.